Uh, Good afternoon, everyone. It's with a big pleasure that I welcome here the Dr. Melissa Ingala to give a talk of today afternoon at 1 o'clock. Melissa is a mammalogist and a microbiologist who uses gut microbiomes to understand the ecology and evolution of bats. She uses molecular techniques to investigate how gut bacteria provision bats with nutrients missing from their diets, and is broadly interested as well in the role of gut bacteria in generating ecological diversity in vertebrates. So she's going to be talking about microbiome functional enrichment across independent dietary transition in bats. So it's with a big pleasure that I welcome you, Dr. Melissa Ingala.
Thank you very much for that wonderful introduction, Dr. Oliveira. I also want to thank the students of the Universidad Federal do Paraná for having me. Sorry, go ahead. Okay. For having me to give this talk today. It's my pleasure to share with you a piece of my dissertation research that I accomplished at the American Museum of Natural History, but I'm currently a postdoctoral fellow at the National Museum of Natural History in Washington, D.C. So today I'm going to tell you a story about how the gut bacteria that live inside of bats actually help facilitate their ecological lifestyle. And I use molecular methods to investigate this, so it's a really good topic for your class here with Dr. Oliveira. But before I jump into kind of the nuts and bolts of this project, I just want to give you some background on natural history museums and how they factor into using these molecular methods to study animal ecology. So I did my PhD at the American Museum of Natural History, and the first time I went to the AMNH was probably in this photo from 1999. And I fell in love with the institution from the very first time I walked inside. But what I didn't know back then was that just a couple of years later, natural history museums would become a foundational part of my research and my professional career. So I did my PhD at the American Museum of Natural History working on the gut microbiomes of bats, and I spent 4 years there. So for the uninitiated, people who may not have worked in or with natural history museums before, I just kind of want to give you a window into this world. So the purpose of most major natural history museums is to document and describe the incredible and vast biodiversity of life on Earth. This picture that you're seeing is one of the halls. It's called the Hall of Biodiversity at the American Museum of Natural History. And you can see that natural history museums are interested in life in all of its facets. Invertebrates, vertebrates, we have crustaceans, mammals, just anything that's alive on Earth, it falls within the purview of a natural history museum. And the way that natural history museums achieve this goal of documenting and describing biodiversity is typically through the use of biological collections. So this is a window into what the biological collections at the American Museum of Natural History look like. This is the ornithology, so the bird collections. And what you can see here is that all of the specimens in a natural history museum are organized taxonomically. So we keep the same species together. And within our collections, you're going to see a lot of things like this. A drawer filled with prepared specimens, organized according to the same species, that you can use to study the evolution and natural history of those animals. But apart from the specimens themselves, one of the really unique things about natural history collections is that each specimen comes with all of this fantastic metadata, which tells us something about where the animal lived, where it was collected, what kind of habitat it lived in, when it was collected. So this particular bird specimen from the Carnegie Museum, you can see, was collected almost over 100 years ago. And also some basic morphological details, what color the feet were, maybe a couple of morphological measurements, things like that. But natural history museums are actually more than just dusty drawers filled with dead things. In fact, most major natural history museums today also archive tissues that can be used for molecular or genomic analyses. So you can see here, I'm busily extracting DNA from fecal material that I collected along with voucher specimens for the American Museum of Natural History. And we can use these molecular tools to give us even deeper insights into the ecology and the evolution of whatever animals we may be interested in. So I've already explained that the goal of natural history museums is to document and describe biodiversity, but something I want you to notice from this picture, which you've seen before, is that these branches of the tree of life actually only represent a small part of the overall biodiversity of life on Earth. Because the truth is that not all biodiversity is even visible to the naked eye. In fact, most of the species that remain to be discovered and described in the world are microbial, things like bacteria, viruses, and protists that are too small to be seen without the aid of microscopes, and thus are underrepresented in the public-facing halls of most natural history museums. But even further than that, the animals that we do focus on at natural history museums are themselves colonized by a diverse array of bacteria, viruses, and protists, and we refer to these communities as the microbiome. So that is actually my major research focus, is using the microbiome to understand something about how animals evolved and ecologically radiated. So knowing that natural history museums do not typically focus on these animals, you may wonder why I would choose to study the natural history of these host-associated microbiomes, especially when natural history museums are not usually focused on them. So I want to give you a brief overview of microbiomes and how I think they address three major overarching goals of evolutionary biology that we do focus on at natural history museums. The first is basically basic discovery and description of biodiversity. As I mentioned, the vast majority of undescribed and undiscovered species on Earth are actually microbial. So if the goal of a natural history museum is to document and describe, there are tons of bacteria, protists, and viruses waiting to be discovered. The second goal addressed by this kind of research is that we can document patterns related to the evolution and natural history of the animal hosts that these communities of microbes colonize. This is a rapidly expanding area of research, but we've really only scratched the tip of the iceberg on understanding how these processes have played out over evolutionary time. And finally, we can turn our view outward and understand the processes and forces that continue to structure these host-associated microbiomes today, including threats to the hosts themselves, such as habitat destruction and land conversion. So if these microbiomes are really important for their hosts, they may have implications for the conservation biology of those hosts. So I'm a mammalogist by training, as Dr. Hernani mentioned, and so I knew I wanted to do this kind of research on mammals. And it turned out that back when I started my PhD 4 years ago, for most mammal groups, gut microbiomes were pretty much undescribed for many, many groups. So I want you to have a look at this bar chart here and see that the distribution of microbiome research effort is not actually distributed evenly across all the major orders of mammals. This is put even more into perspective when we consider what groups of mammals actually make up the majority of the diversity of mammal life alive today. So have a look at this pie chart here. Rodents and bats alone make up 60% of all mammals, meaning that all of the other mammals comprise the other 40%. But you can see here on the bar chart that primates, which compose just a tiny sliver of mammal diversity, actually take up almost 30% of all microbiome research effort. And so I knew I wanted to focus on a somewhat neglected group of mammals, and that is why I chose to study bats. So you may be asking, well, what are so special about bats? First of all, there are quite a few of them, so it's not hard to find species to focus on. With over 1,400 described extant species of bats, they make up 20% of all living mammals and are unique in being the only mammals capable of true powered flight. Bats have an incredible morphological diversity, which this is just a little snippet of that diversity, ranging in body size from the smallest living bat, the bumblebee bat, weighing in at a whopping 2 grams, so that's about the same as a US penny, all the way up to the giant golden-crowned flying fox, which weighs in at 1 kilogram and has almost a 2-meter wingspan. So just an incredible diversity of body sizes in this group. But apart from morphology, bats are actually some of the most ecologically diverse mammals on Earth as well. They've radiated to fill every terrestrial feeding niche, including nectar and fruit feeding, insectivory. There are carnivorous bats, as you can see in the top left. And perhaps in the most interesting transition, there are 3 species of bats that feed on blood, and which do occur in Brazil. So the distribution of these ecological niches is shown here on this phylogeny, which is basically a family tree of the bats. So of the 1,400 described species of bats, about 70% of them are insectivorous, which are represented by branches colored in tan on the tree. Others have radiated to become nectarivorous, shown on the tree in pink. And actually, there are quite a few plants which are primarily pollinated by bats, including the tequila agaves, shown here. In addition, we have a bunch of fruit-feeding bats that have evolved several times across the tree, including this adorable pteropodid munching on a grape, it looks like. And our carnivorous bats have evolved a few times on the tree, shown in brown as well. You might be able to make out in this GIF that this bat is actually chowing down on another bat. And then we have our 3 species of blood feeders shown in red on the tree. This particular feeding ecology has only evolved 1 time in 3 very closely related species. Don't be fooled by the close-up, by the way. Vampire bats are pretty small. They only have about a 6-inch wingspan, so they're not as big and scary as this GIF might lead you to believe. You're probably safe from a vampire bat if you're not a cow or a pig. So I mentioned that they have this incredible morphological diversity as well. And traditionally, this has been one of the tools that's been used to study how bats have adapted to their unique diets. So I'll give you this example. On the left, you can see the nectar-feeding bat Mucinicterus harrisoni, which has an elongated rostrum, which likely evolved to help it access the nectar that is contained deep within flowers. In the center, we have a kind of generalized bat morphology, well-suited for insect feeding. And on the right, we have the adorable face of Centurio senex, which is a fruit-eating species that has evolved a very short rostrum capable of generating high bite forces, which it needs to feed on the hard-seeded fruits that it has adapted to feed on. But this morphological adaptation to diet actually only deals with handling and processing food. It doesn't tell us much about what it takes to nutritionally be able to specialize on these interesting food resources. So how do bats actually digest and process that food? This is one of the questions that motivates my microbiome research. And I'll show you how I think of this system as potentially occurring. So when a bat eats a food item such as this plant, the nutrients from that food are used to generate its own metabolic activities, and the bat breaks down the nutrients in that food and absorbs it for its own metabolic processes. But the same nutrients from that food also serve as a substrate for all of the microbes that colonize the gut of that bat. And these microbes do their own metabolic processes, and in the process of doing that, they liberate nutrients that can then be secondarily absorbed by the host, therefore supplementing the host's own metabolism. And so knowing this, I came up with a question of, do bat microbiomes actually provide essential nutritional functions to their hosts? In other words, do bats depend on their gut bacteria to actually help them be able to specialize on these food resources, especially ones that are nutritionally challenging, such as fruit, blood, and nectar? So as I mentioned, microbiomes are not typically collected or archived in most major natural history museums. And because of that, I had to go to the field to collect new, fresh material myself. So during the course of my PhD, I took 4 separate trips to 2 different countries to do microbiome fieldwork, expeditions, and collecting. But most of the data that you'll hear about in this talk today came from Belize. So let me introduce you to Belize. It's a small country located in Central America, bordered by Mexico to the north and Guatemala to the west, and the Caribbean Sea to the east. And the site that we work in, called Lamanai, looks a little bit like this. It's a seasonally dry tropical forest, just a really beautiful place to work. And fortunately, we are able to net for bats in a protected reserve called the Lamanai Archaeological Reserve. This tract of land is several hundred acres in size, and it protects the 3,000-year-old ruins of the Maya city of Lamanai. At this point, I just want to stop and acknowledge the Yucatec Maya people, who are the traditional inhabitants of this land and still are the main custodians and carers for the land today. We work closely with many people from the adjacent village to Lamanai Archaeological Reserve, and we're always very grateful for their help, support, and knowledge in these expeditions. A few kilometers away from Lamanai, we have a secondary site called Kakabish. Kakabish is more of a degraded forest fragment. As you can see here, it's even cut in half by a road. But bats do live here, and it's actually a great place for us to go and target certain species like vampire bats. Kakabish, unlike Lamanai, is not quite as protected. However, underneath the forest that you can see in this photo are yet more Maya ruins, which are in the process of being excavated and protected through the Belizean Archaeological and Cultural Ministry. Across the lagoon from Lamanai is actually a nice piece of dry tropical savanna where we also are able to net for bats, and we sometimes get different species here. So this area has a real heterogeneity of habitats, and all of them are really great for catching bats. Even the rivers and tributaries themselves are really fruitful bat netting habitat. And in particular, when we're targeting species that feed on fish, we try to at least arrange one trip to go out and target them using nets placed across the river so that we can catch them as they come in to trawl for fish. The fieldwork aspect of catching bats involves two kind of workhorse tools in the trade. On the left, you see a mist net, which is a fine gossamer nylon net, which can be placed out and opened. And the bats actually can't see it because it's so fine. People can't either. I walk into these things more often than I would care to admit. But they work really well. So the bats fly into the net, they get tangled. We check the nets every 10 minutes or so and untangle them so that we can take them back to the lab to take measurements and samples from them. The other tool we use is shown on the right. It's called a harp trap. It's called that because it superficially resembles a harp. And the way that the harp trap works is that it has 3 panels of fishing line, basically. And the bats fly into the fishing line, pivot to avoid the first layer, but usually hit the second or third layer. And then they tumble down into the soft canvas holding bag where they're protected from the rain and from predators. And then we come and collect them and also bring them back to the lab. So we use these 2 different tools to capture bats because you actually capture different species in mist nets versus harp traps. And so by using them both in combination, we can get a pretty good sampling of the taxonomic and ecological diversity in these regions. So as I mentioned, we untangle the bats from the nets and we put them in their each in their own individual little holding bag, which are shown here on the right. And so we kind of let them hang out in those bags and take them back to the lab for processing. And so this is a time-lapse video of our central processing lab in Belize. And as you can see, everybody kind of works together to collect whatever samples they need from those animals. After the bats are finished being sampled, measured, what have you, we usually release them back at the site where they were initially captured on the same night. So once the fun part, the field work, is over, we have all of our samples and we have to take them back to the lab for processing and data generation. So when I'm in the field, the main sample type that I collect to do microbiome work is fecal material, as you can see here. And so I'll just give you a really brief primer of some of the molecular methods that we use to study host-associated gut microbes and how these projects play out. So once you have your fecal sample, we preserve it in liquid nitrogen, take it back to the lab, in this case, the American Museum of Natural History, and we extract the DNA from the fecal sample so that we can use it for molecular library preparation and sequencing, get the data from that, and then apply bioinformatic techniques to actually analyze that data. So from your fecal sample, when you do the chemical DNA extraction, you don't just get microbial DNA out of it, you get bulk DNA, which means it's a mix of microbial DNA, host DNA, and DNA from whatever dietary items the bat might have eaten that night. So our goal is to be able to selectively amplify just the microbial DNA from this bulk DNA isolate. And the way we do that is by using PCR to amplify only bacterial 16S genes. 16S is a really useful bacterial barcode that can be used to understand what bacterial species are present in a sample and at what relative abundance. So by using this selective amplification, we're able to generate what are called 16S DNA libraries, which contain only bacterial DNA and minimal host or dietary contamination. So once we have those libraries, we're able to sequence them on the Illumina MiSeq platform. And the data that we get off the machine looks a little bit like this. So this is a standard FASTA file where you have all of your sequences for each read that was processed on the machine. And as you can see, it's kind of a jumble of A, C, T, and G, which are, of course, the bases that make up the DNA code. But we can't really say anything much about what bacteria these are or what they might be doing in this format. So we have to take that raw data and then process it through these bioinformatic pipelines, which can be used to assign taxonomies to the bacteria, so who are they, as well as functions to tell us something about what they're doing. Now, I mentioned that 16S is a bacterial barcode, and I really mean that because much like a barcode that you might find on an item at the supermarket, it tells you a little bit of something about what that bacteria is, but it doesn't tell you directly what it does. So just as you might scan a barcode and get a little SKU number telling you the item name, that doesn't really tell you what you can do with the item, versus if you're able to read the box, you know that this is actually cake mix. So how do we go from bacterial barcode to bacterial function? To do that, we have to use a somewhat complicated prediction algorithm that I'm going to simplify just so you understand how we get from point A to point B. So the way that this algorithm works, it's called Crust. The algorithm has sequenced genomes. These are bacteria that have been brought up in culture, whose genomes have been sequenced entirely and functionally annotated. So we know exactly who those bacteria are, and we know what they do. So using these sequenced genomes with known functions, we're able to create a reference phylogeny, or a reference family tree. And we add our samples, which are shown on the tree in blue, to the phylogeny where we think they might go based on their closest relative. From that taxonomic and phylogenetic placement, we're able to generate predicted functional profiles based on how close our bacteria from the bats are to these sequenced genomes whose functions we know. So the output from this program is a bunch of bacterial metabolic pathways and relative counts that have been scaled and corrected for 16S copy number. Since some bacterial species have more than one copy of 16S, that's really important to do. So for the total data set that we used for this project, we had a wonderful number of taxa, 60 species of bats, comprising all known terrestrial dietary niches. And we combined our data that we collected in Belize with previously published data from bats from the Afrotropics. So we actually even had 2 biogeographic regions represented. And in addition to that, we actually have what are known as repeated dietary shifts. So I'll show you, for example, fruit eating in bats has evolved at least 2 times, and we can see that on the tree here. We have our old world pteropodid fruit eating bats and our new world leaf nose fruit eating bats, which have been separated by millions of years of evolution. So we're pretty certain that these 2 groups have evolved to eat fruit independently of one another. This is a really important consideration because many times animals have correlations between their dietary ecologies and their phylogenies. What that means is that closely related species tend to also share ecological traits. And so it can be difficult to parse whether the patterns we're testing are due to shared evolutionary history or ecology. In this case, we can uncorrelate those 2 variables so we can actually tell which one is driving the influence. So this looks like a somewhat scary graph, but it's not. I'll break it down to you. This is the results of our functional profiling analysis. So the first thing that we found here is that functions stratify by feeding niche or guild. When you're looking at this plot, the important thing to take away is that each dot represents an individual sample, and its placement on this figure is determined by its similarity to other samples within the data set. So two dots that are very close to each other in this ordination space have very similar microbiome functions, whereas those that are farther away have different microbiome functions. So we're able to apply a statistical test to this type of data called PermANOVA. It's a permutational ANOVA. And from that test, we were able to determine that the feeding niche to which each bat belongs is a significant predictor of what its microbiome is doing. However, the important thing to kind of take away from this figure is that there's a lot of overlap here. And we think that's because a lot of bat microbiomes share what are called housekeeping functions. So this means that whatever you eat, you basically have a basic standard toolkit of microbiome functions that is not going to be impacted by your particular dietary lifestyle. However, there must be some bacterial functions that are driving the changes that we have detected by this statistical test on this data. In order to find out what those actual bacterial functions were, we had to apply a different test. This test is called LDA-LEFSA, and I'm not going to go into the details of it, but basically it uses an LDA-type test to determine which functional pathways are significantly differentially enriched between two groups. So in this case, the first thing we wanted to understand is kind of higher level ecological differences. So what are the bacterial functions driving the difference between plant-feeding bats and animal-feeding bats, which includes carnivores, insectivores, and blood feeders. So from this analysis, we found that plant-feeding bats have significantly enriched essential amino acid pathways. So these are pathways that are involved in generating amino acids that vertebrates are incapable of generating themselves. Those amino acids either already have to be present in their diet, or they can be synthesized by bacteria, which contain all of the essential amino acid synthesis pathways. So that's what we found for the plant feeders. They also had some enrichment in things like glycogen synthesis. So this is the fat that animals generate and burn during physical exercise, and sugar degradation, which makes intuitive sense because we know that plant-feeding bats are primarily fruit and nectar feeders, so their diets contain a lot of sugar. On the other hand, our animal-feeding bats had a much more generalized suite of functions, including fermentation, fatty acid synthesis, and cofactor and vitamin biosynthesis. So this is, like I said, a very high-level ecological distinction. You know, it's binary between plants and animals, but we know from the intro to the talk that bats actually feed on much more diverse food sources than just plants or animals. So we actually broke it down into their more fine-scale ecological niches, and we found a similarly diverse array of functions that define bats according to their feeding niches. So for example, everything you see in blue here is a pathway that LDA-LEFSA identified as belonging to or defining blood-feeding bats. And the same goes for insectivores, frugivores, carnivores, what have you. So I just want to zoom into one of these pathways so we can talk a little bit about the metabolic significance of what these bacteria are doing. So in the fruit-eating bats, one of the pathways that we found to be differentially enriched is L-tryptophan biosynthesis. So L-tryptophan is a precursor for generating tryptophan, which is a conditionally essential amino acid. And the way that you read this graph is that each bar represents an individual bat, and the height of that bar represents the relative abundance of that bacterial pathway in that animal. So within the groups, frugivore, insectivore, and the vampire bats, the median line, so the median enrichment across the entire group, averaged amongst all individuals, is shown by the hashed line. And so you can see that the fruit-eating bats have a much higher enrichment of this L-tryptophan pathway compared with insect-feeding and vampire bats. So instead of showing you a whole bunch of those graphs, I've just kind of condensed them into more intuitive language. And so the other things that we found to be enriched like this were other essential amino acid synthesis pathways, including valine, leucine, and isoleucine pathways. We also found starch breakdown pathways to be hyper-enriched in the fruit-eating bats, and secondary metabolite breakdown pathways. Secondary metabolite breakdown is actually a very interesting one to me, because plants actually produce a lot of secondary metabolites that are produced to discourage herbivores from feeding on them. These are things such as tannins, which are the compounds which give wine its kind of astringent flavor. And so it's interesting that these fruit-eating bats would have bacteria that are able to help them handle and break down those secondary metabolites found in their plants. And in general, all of these functional pathways make good intuitive sense when we understand that the fruits that bats eat are very variable in their nutritional composition, but compared to things like insects or vertebrate prey, they're pretty low in protein, fats, and vitamins. And so the bats might be relying on their bacteria to help kind of bridge the gap, nutritionally speaking, between the amount of protein, fats, and vitamins they need to survive and what's available for them in their diets. Similarly, we found that vitamin biosynthesis is enriched in vampire bats, and so this is just one of those functional pathways. In this case, this is thiazole biosynthesis. Thiazole is a precursor for making certain classes of B vitamins, which are essential nutrients that all vertebrates need to live. And so it's interesting to see that these vampire bats actually have almost 3 times the average enrichment of this functional pathway compared to insect feeding and fruit feeding bats. In addition, nucleotide and nucleoside degradation pathways were also enriched in vampire bats compared to these other feeding niches, as well as vitamin cofactor biosynthesis pathways and nitrogen compound metabolism. All of these pathways, again, are consistent with the nutritional deficits that exist in blood. So blood is about 92% water, and the 8% remaining is all of the cellular components of the blood, which tend to be pretty low in vitamins and lipids as well. So it seems like vampire bats are really relying on their microbiomes to provision them with certain kinds of vitamins and nutrients that are totally missing from their blood diet. An interesting thing is that a previous study, which integrated studying the genomes and the metagenomes of vampire bats, found similar results to what we present here. So in particular, they found that vitamin and other nutrient metabolism pathways were upregulated both in their microbiomes, shown by the keywords in red, and in the genomes of the vampire bats themselves, shown by the blue gene names. Similarly, they also found that nitrogenous waste handling was upregulated, as well as amino acid metabolism as well. So our results are really consistent with this, but we've expanded it to include other dietary niches apart from just blood feeding. So one thing I'd like to mention is that these results rely on the quality of the information about what the bats actually eat. So in order to classify something as a frugivore, you have to have some idea that it's actually eating fruit. But for most species, we actually lack pretty detailed dietary information about what these animals are actually eating. Bats are small, nocturnal, and they fly, so directly observing their diets can be pretty tricky. So for curiosity's sake, we decided to also profile the diets of these bats using a combination of barcodes to see if detailed dietary information could change the inferences we're able to make about microbiomes. So we use the following primers to selectively target invertebrates, plants, and vertebrates. And from this data set, we profiled the diets of 25 species of bats, resulting in over 800 dietary molecular OTUs, which you see in this bipartite network have been collapsed up to the level of taxonomic order. All the bats are listed on the left in this plot, and their connections to the plant and invertebrate species located on the right are scaled according to the number of observations. So in this case, a thicker bar represents more observations between that bat and that diet taxon. And so when we summarize these interactions as community submodules, we find some pretty interesting patterns. So in this plot, each box represents a submodule of the total community of bats, shown on the left, defined by their resource use, shown on the bottom axis there. And so these are species that we had previously just been classifying as either frugivores, insectivores, or carnivores. And if you actually overlay those icons on top of the modules that we identified using these molecular techniques, you could see that they're not very discrete. So there are some modules that are only feeding on plants, but there are modules that are kind of a mix of plants and invertebrates, or a mix of vertebrates and invertebrates. At least at this site, we can summarize our community interactions this way and get a much more fine-scale picture of what bats are actually eating. The interesting thing is that when we rerun those microbiome analyses using our diet modules that we actually directly assessed, it improves the explanatory power of our Permanova test by nearly 10%. So if you actually understand better what these animals are eating, you can explain a lot more about how and why their microbiomes are functionally variable. So to summarize these kind of major bullet points, bat microbiomes seem to be functionally enriched in pathways that are potentially really adaptive to their diets, particularly those species which feed on nutritionally tricky foods to specialize on, such as fruits, nectar, and blood. So these results actually imply that bacteria contribute to really important metabolic processes in their hosts. And disruption of these processes could have really salient impacts on host health and fitness in the wild. And then, the traditional guilds that we use to describe these animals are, of course, useful, but having more detailed dietary information better explains the functional variation we can observe in these gut microbiomes. So I hope by the end of this talk, I've convinced you that host microbe interactions add this rich new layer to our understanding of global biodiversity. And this kind of work actually spurs a lot more questions than it answers. So we know that microbiomes are tightly linked to host ecology, but there are millions of directions that we can take this research. How are microbiomes involved in host aging? How do these metabolic interactions actually constrain or enhance host fitness? What do microbiomes contribute to host health, and how is that impacted by conservation concerns? And how do microbiomes really mechanistically contribute to host metabolism? All of these are open questions that we are in the process of following up on. And for my current postdoctoral fellowship at the Smithsonian National Museum of Natural History, we're trying to follow up on this project by really asking how host genomes and metagenomes collaborate to allow bats to specialize on the unique foods that they do. So with that, I have a lot of people to thank for this research. None of this could be possible without all of the people listed on the screen and the many sources of external funding that we secured to be able to pay for the molecular parts of this research and the fieldwork. And with that, I just want to thank all of the students and viewers for tuning in today, and thank you for your attention. And now I think we are going to move to a Q&A session with the students in Google Meet. So thank you all. Enjoy the rest of your day and have a good one.